Today I'd like to talk to you about the idea of using DNA sequencing technologies, also known as genomic sequencing, in newborn screening programs.
But first, let's talk about what DNA is. DNA contains the instructions that tell our bodies how to grow and develop. Each person has a unique DNA sequence that affects many things about them, such as their eye and hair color, as well as their chance of developing some diseases. DNA is made up of a sequence of letters, A's, T's, C's, and G's. So what is genomic sequencing? Genomic sequencing is a technique that is used to read DNA. It finds the order of the letters of a person's DNA one by one. This DNA sequence can be referred to as genomic data. There are a lot of letters to be read, which means that genomic sequencing can produce a lot of data. Once the DNA sequence is obtained, the genomic data can be used by doctors and scientists to help identify the causes of genetic disease in patients. Doctors and scientists look for changes in the DNA, called variants, that are different between people with diseases and people who are healthy. These changes might be very small, like spelling mistakes, or bigger, where large runs of letters are missing.
Now let's talk about what newborn screening in Australia looks like. Newborn screening is a government-funded public health initiative that aims to detect rare and serious childhood onset conditions in babies before they start developing symptoms. Earlier detection of these babies means treatments can be started earlier, which leads to better health outcomes. In Australia, newborn screening programs screen for approximately 25 conditions, although this varies a bit depending on which state or territory you live in. Newborn screening is often referred to as the heel prick test because that is how drops of the baby's blood are collected in the first couple of days after they are born. A nurse or midwife normally does this, either in the hospital or at home. The nurse or midwife also gets consent from the parents to take the blood sample. The samples are tested by a laboratory. Abnormal biochemical levels in the blood indicate that the baby has the early signs of one of the serious but treatable conditions being tested for. Each condition has its own biochemical that is tested. If an abnormal newborn screening result is found, parents will be notified by a health professional and further tests will be done to confirm the results. We are proposing to use genomic testing in newborn screening programs. This would probably be in conjunction with the existing biochemical testing. We want to know how you think this should work.
Thanks for listening, and we look forward to seeing you in the focus group.